Hello everyone, and welcome to the first episode of Medicool, brought to you by the Technion Faculty of Medicine. My name is Roberto, an MD PhD student at the Technion, and today we dive into a very interesting subject with a lot of buzz around it, the microbiome. We are joined by our own Dr. Naama Geva-Zatorsky, head of the Microbiome Research Lab from Rappaport Technion Integrated Cancer Center at the Technion Faculty of Medicine, and my own PI. Hello Naama, and thank you for joining us to talk about science.
Hi, Roberto, and hi, everyone. I'm really happy to be here.
Naama, you've had quite the interesting academic journey on your way to becoming a PI at the Technion, finishing a dual degree in chemistry and biology at the Tel Aviv University cum laude, continuing from there to the Weizmann Institute of Science to your master's and PhD studies, and off to Harvard University to pursue a postdoc at the Casper Lab, where you first started studying gut bacteria and their effects on their human hosts. You're also an entrepreneur and co-founder of BiotaX, all this while being a mother to 4 children. A hard path, no doubt. Can you please tell us more about this journey, the highs and lows, and your take on it while looking back?
Yes, of course. So I can say that throughout my journey, I always followed my heart and my gut feelings. And you'll hear more about the gut feelings soon. So for my bachelor's, I really tried to convince myself to study a topic, a profession like being a lawyer or something around that direction. But I followed my heart and I went to study biology and chemistry. So my bachelor's was in Tel Aviv, at the Tel Aviv University. I always loved biology, chemistry, science, always looked for science and behind the house of my parents in the fields. So I loved this choice. During my bachelor's, I was intrigued by molecular biology, and I did a few projects there at the Tel Aviv University on molecular biology. And also, I always probed for opportunities, and that's how I learned about the summer program at the Weizmann Institute. And in one of my summers of my bachelor's degree, I went to the Weizmann Institute for a couple of projects there. And I stayed there for my master's and PhD in Ori Alon's lab, who is a world pioneer of systems biology. So from Ori, I learned how to combine physics and biology and look at biology with some computational aspects. And we studied the dynamics of proteins in cancer cells. However, I always loved microbes, and I followed this dream for my postdoc in Dennis Kasper's lab at Harvard Medical School in Boston, where I spent 6 years to study how these gut microbes communicate between themselves and how they complement our functions, and mainly our immune functions. I really enjoyed that time as well. During my postdoc, I was in touch with the Technion. I was always looking up at the Technion, and I was very happy to be accepted to the Technion, and I'm really happy with this opportunity. I opened my lab in 2017, and since then it's history. So I love my lab. I love you guys, my students, and I really feel it's a second home, and calmness, fun, and a lot of excitement.
That's amazing to hear about this scientific journey of yours, even though I just noticed that your PhD and master studies were quite different from your postdoc, even though that now we know that the microbiome is being integrated to various topics and various fields of research. So can you tell us more about the microbiome, what it is, how does it affect us, and maybe just elaborate on the research you conduct in your lab?
Of course. So first, on that point that you raised, I agree. The topics, if you read even the papers, they will look very different, but I really encourage the students in the crowd and any person that listens and is interested in research to probe different types of research, because it's really encouraging and also enriching, and it makes the research even better if you combine several disciplines. So the microbiome, yeah, it's a collection. It's the collection of organisms that live in and on us. We have actually the same number of microbes to the number of human cells in our body. So we can look at ourselves as walking microbes, living microbes. And we realized in the past few decades that they mean a lot to us. So the first breakthrough, or the major breakthrough that started this renaissance of the microbiome field was about 2 decades ago, when the deep sequencing machines were developed. So now we can sample this wall, our sweatshirts, or our stool, which is our favorite sample. And we can just throw it to this machine and sequence. And from these sequences, we can analyze which types of microbes are present in this stool or on our skin or wherever. And what this helped us to understand that the microbes alter in health and disease, and that raised a huge amount of research, realizing that they are important to our health.
Wow, this sounds a lot like science fiction, but can you elaborate more on how do they do that? How do they interact with our body? Do they interact with our immune system? Do they interact with the food we eat? Like, how can we say that these microbes actually affect our lives?
Yeah, so all the answers are correct. So we need these microbes to digest our food. There was actually a really cool story about babies. So the mother, towards the end of her pregnancy, acquires a certain microbe, which is transferred to the babies. Actually, they have very, very nice names like Bifidobacterium. And without this microbe, the baby cannot digest the mother's milk. So the mother is investing a lot of energy to create this complicated milk that the baby cannot digest without this microbe. But the microbe is not there only for that. It helps the baby to digest the sugars in her food, but also it helps the immune system of the baby to develop. So that's one really cool example about how these microbes can help us digest our food, and they're really there to digest many types of the food we eat. And actually, we can use the content of these microbes to understand both on diagnostics and to understand about which food we eat. Like, you can give me your stool and I probably could tell you if you're a carnivore, an omnivore, or a herbivore.
Well, sounds like they're implicated in every aspect that we might imagine. But so now our perception towards microbes have been shifted. Like, now we know that they're not all bad microbes, rather some of them are quite beneficial, and we need them for good development and to be able to do various functions that we cannot be doing without these microbes. But how can we use our knowledge in the microbiome research field to intervene with patients to limit their disease or to better their lives? Like, we've all heard about the probiotics and various food supplements, but how can we implement this to a bedside manner to better the lives of patients?
Great question. So believe it or not, there are treatments of the microbiome in the form of fecal transplantations. So exactly as it sounds, treating sick people with feces of healthy people. So the sentence, "your poop is our gold," totally speaks to us. However, we and others aim to understand what is it exactly in the feces that treats diseases, and to develop more accurate therapies based on the bacteria themselves or their products. Perhaps it will be easier psychologically to treat people with the molecules of the bacteria rather than the bacteria themselves, or let alone poop. So to do that, we developed also a technology that allows to tweeze out bacteria of interest in their live form from natural samples, such as, you can already guess, poop. And based on this technology, we spun off a startup company called BiotaX.
A poop pill. I hope it doesn't smell though. But like, it sounds to me like you're taking it towards a more precision medicine approach, and this is actually have been a great. It's been a very prominent field in the biomedical industry lately, and also in the medical research, both clinical and preclinical. So can you tell us more about this cooperation between the academia, basic research, clinical research in the hospitals, and biomedical industry? How do they cooperate and how are they different?
Yeah, so things have really evolved lately, and I really believe that the biomedical industry and medical research are very intercalated between each other, and they should be even much more involved one in the other. And we have to protect the basic science, that's for sure. But the biomedical industry has also become research institutes in a way. So the research in the biomedical industry is cutting edge, and the research in the academia is cutting edge. It's true that in the biomedical industry, it's more directed towards a goal. Also in the academia, it's directed towards goals, but it's a little bit, in my view, more open and more wide, which is great, and that allows also serendipitous discoveries many times. And I'm now lately in the 8400 Health Network, which is exactly built towards bridging the gap between industry, academia, and hospitals and medical institutes. And I really think that should be a goal of all of us, because by lowering the barriers, we can lead to better research, both in academia and in industry, and get to better lives of the patients, which is the goal of all of us.
This sounds amazing. Like bridging the gap between these two bodies has shown itself to be very beneficial towards the mutual goal in the end, like you said. But I would love to hear more from you. How did you go into the world of startups? Like, you're one of the few researchers that I've heard about who had the courage to go into the startup world and take a leap and open up a startup. Can you tell us more? How was it? What was the process? What new challenges did you face? And how did you overcome these challenges?
So yes, it's a great question. It actually wasn't a goal or a plan of mine to have a startup spin-off of my lab and to open a lab. And actually the startup came quite early in the academic career. So, however, it did come quite naturally when the Technion has encountered me with the entrepreneur Shai Hillel, with whom we co-founded BiotaX together with Dean Light, who is also actually a Technion graduate. The first meeting with Shai was very positive and exciting. We understood that we'd want to do something together and brainstormed about options that can arise from the research in my lab. The bacterial isolation technology was a really good fit, and the rest actually is history. In BiotaX, we have pursued several clinical trials, including in colon cancer and in lung cancer. We're collaborating with MDs, and we're taking several interesting directions, all regarding the microbiome, mostly in health, and also regarding the environment.
So it seems your startup is actually manifesting the cooperation between different entities like hospitals and MDs, basic research and academia, and the biomedical industry. But how do you see this cooperation continuing in the long run, like 5 or 10 years ahead? Do you see it getting stronger, more intricate, or do you see their paths diverging again?
Wonderful question. I vote for the former of the options that you gave, that there will be much more intercalated in between each other, and the barriers will be lower. And I think it will, both industry and academia will make bigger leaps forward, more towards patients, more towards applications, if we will talk to each other much more and find ways to reduce the barriers, have less secretive junctions and more openness, more modes of openness that will still protect all the sides, but find ways to talk freely, and that will help us to move in much bigger steps forward. And I really think this is crucial, and that's how I see academia and medical industry in the future.
That's amazing. Actually, quite a relieving answer, I think, for her. Most of us who hope that this cooperation continues for everybody's sake. But I would also love to hear from you. What's next? What's next in the microbiome field? What are you doing and working on in your lab now? How are you planning to move forward?
Yes, this is the topic that I love to talk about the most. So thanks for this question. In the lab, we now more and more understand that microbial names are less important and their functions are much more important. It's actually both in the lab and in the whole microbiome field in general. And specifically in the lab, we're interested in mechanisms that allow them to function. So function means like upregulating or downregulating an immune cell, meaning like helping us to overcome an infection, let's say, or helping us to reduce inflammation.
So bacteria itself can help us reduce, our susceptibility to other bacteria.
Yeah, it's a great point. Yeah, exactly.
That's
so
amazing.
The bacteria can fight other bacteria, and the bacteria can also help us recover from diseases that are not necessarily related to bacteria. And these are the functions that interest us the most. So we're interested in the mechanisms. It's like the wiring of the bacteria that enable them to pursue these functions. And not only that, we realize that these functions can be altered. So like switches of lights that can go on and off, we are now studying these switches in the bacteria. So how they can change their functions in different conditions. So conditions can be depending on whichever other bacteria are next to them, or whether there is a disease now or not. We see that their functions are altered, and that's what excites us the most. When are they altered? Why are they altered? Can we manipulate them? And last but really not least, we are also studying the viruses of the bacteria. They're called bacteriophages. So we know that we can have the flu or viruses, and mainly in the winter, as people. But also the bacteria can be infected by very, very tiny viruses. So we're very excited by these interactions. Especially how these viruses can change the microbial populations and even their functions. And these are actually the researches that are ongoing while we're talking in the lab.
Well, it seems like we have a lot of research to do in the microbiome field. Each time that we find something, another thing comes along that sort of opens up a whole new field. This is actually one of the reasons I was interested in the microbiome, and it sounded like it's a lot like science fiction again, and this is actually amazing. Apart from the great academic achievements, you're also a mother to 4 amazing children that I've personally met. But how do you juggle all of this? How do you keep the work-life balance going on?
Yeah, so exactly. You personally met them and you're in the lab. So I combine them also with the research. So they hear a lot. They know what is a grant. They know what is a paper. And I talk a lot about work at home. I think they're happy to hear about the research and the work. And they know what a deadline means. And they know that now they need to help me work. Yeah. And I bring them a lot to lab, even on weekends many times. And
which is a great addition, actually. When you bring them, they're amazing. Before we depart, would you like to say a few words to our listeners?
Sure. Thanks a lot for inviting me. I really enjoyed talking with you. I really thank you for joining my lab. And I'm very happy with the collection of students in the lab. I have to say that we do have a really interdisciplinary research in the lab with students that come from computational backgrounds and students like Roberto who come from MD studies that combine PhD research during their MD studies, and clinicians. So it's really, really fun. And I encourage this interdisciplinary to everyone. And thank you so much for this time.
Thank you very much, Naama. And a big thanks to our listeners and the Ruth and Bruce Rappaport Faculty of Medicine at the Technion who make these sessions possible. Stay tuned for our next episode on Medicool podcast.